10 o'clock. Okay, good morning, everybody, and welcome to the virtual research presentations of the second-year cohort of the Carolina Postdoc Program for Faculty Diversity. This program really serves a very important pathway to academia here at UNC, supporting academic departments in all of our schools and the college in their efforts to attract, develop, and hire underrepresented scholars. All 6 of our scholars, the second years, are being recruited for faculty positions as assistant professors here at Carolina that would begin in July of 2022. So today's presentations are just short overviews of their research that they've been working on for the last year and a half. So I'll turn it over now to Joyce Tan, Associate Vice Chancellor for Research, who will moderate the session. So Joyce.
Yep. Good morning, everyone. We have the second year scholars who will be giving short talks today, as Terry had mentioned. We will have a few minutes after each of the talks for questions. So please feel free to type your questions into the Q&A box at the bottom of the screen, and I will read them out, read out your questions. So first, we're going to start with Sean Matharoo, who is from the Department of Romance Studies. And his title of his talk is A Critical Reappraisal of Energy from the Perspective of 20th and 21st Century French and Francophone Literary and Intellectual Histories. Sean.
Hello, I hope everyone's doing well. Thank you for the generous invitation to speak today. I wanted to share with you a quick kind of summary of my research that I've conducted. And some of this stuff is provisional, even the title actually in hindsight. Now I'm still kind of identifying particular starting points, like where should I begin my research. So I'm just going to jump in real quick, and I'd be excited to address any questions or comments afterwards. So responding to the Anthropocenic energy crisis and the need to transition to alternate energy sources, I argue that speculation may contribute to a transformation of what has been referred to as petroculture, which limits our linguistic imagination to oil. Such a limitation reflects the ravenous exploitation of what Andreas Malm has called fossil capital, or a triangulation of capital, labor, and extra-human nature. This exploitation is isomorphic, I argue, with the colonial racial violence that structures the non-white, non-European, quote-unquote irrational, and ultimately non-straight, non-normatively abled, non-male, non-human as the easy, limitless resource of its opposite, the anthropos of the Anthropocene. This violent commodification of energy precludes the development of expenditures of energy outside the capitalist economy of use and exchange. In the energy humanities, the study of aesthetics has been sidelined because it's been argued it lacks use value. My research addresses this lapse by considering how the aesthetics of French language post-apocalyptic literature, media, and philosophy gift us an energy resistant to easy use via the activation of speculation. My research suggests that speculation isn't bound to anthropos, or man with a capital M, but cuts across matter, organic and inorganic. It's a deconstruction of the presupposition that non-humans are irrational or non-sapient. From a subaltern position, I speculate that the apocalypse has already happened, and this is an argument that's been made by several indigenous scholars with which I am in dialogue in my ongoing research. And we might endure its disorienting effects by sifting through its ruins for energy. Speculation authorizes us to approach the energy crisis not from the perspective of the world as it is in dread of extinction, but from an alternate possible world where the apocalyptic future has already come to pass and is approached with openness. Such a departure from alarmism has impelled me to adopt a writing style that functions according to a queer logic of rational systematization by way of quantum leaps or sursauts quantiques, or sudden bursts of energy that perform the pleasurable and painful search for energy outside the capitalist economy and towards something more like a left accelerationist gift economy. I'm updating my doctoral thesis into a book whose updated title is After Yesterday, Toward a Solar Semiotics of Energy. I formalize a new definition of speculation that consolidates G.W.F. Hegel's fabled dialectics of absolute spirit with contemporary French philosopher Quentin Meillassoux' mathematical absolute that is the necessity of contingency. I'm in the process of formalizing a new definition of energy that differs from the everyday definition of it as the ability to do work. Responding to recent theories about dark energy in physical cosmology and astronomy, which it has been argued is driving the accelerated expansion of the universe, I turn to French and Francophone literary and intellectual histories for clarification. Basically, in conducting research on the energy concept, I'm now wondering if its polysemic quality is not necessarily the symptom of a profound confusion, but rather an effect of speculating on the possibility that anything might change in the direction of egalitarianism, justice, and emancipation, or maybe the infinity of words. I demonstrate the energy of speculation with close readings, and this is a provisional list. I'm still struggling to kind of identify an origin point for where the book will begin. But it ranges, and I'm happy to talk about the individual case studies. I'm looking at a French graphic novel, an Algerian speculative fiction novel, a Lebanese-American book of poems, French science fiction novel, as well as in the conclusion, I'm hoping to kind of get a little bit more speculative and kind of gesturing out toward a second project by looking at a variety of different forms of media, including animated films, music, television, film, and actually a video game series that I think is relevant to these questions. So each chapter in the book is going to be designed as a linguistic superimposition or perhaps a virtual simulation of the post-apocalyptic environments these materials depict: the jungle, the tundra, the city, the underground, the desert, the forest, and in the conclusion, animated sonic, celluloid, and digital spacetimes. In the conclusion, I'll formalize the solar semiotics of the book's title. This materialist semiotics is recognizant of the infinite generosity of the sun, authorizing delirious, critical, excessive, and corporeally and affectively intense reading practices and contingent writing practices that free language via speculation, gesturing to solar-powered futures beyond the apocalyptic present and the binary categories that structure it. Such practices, I argue or suggest, perform the sense of wonder constitutive of the science fiction genre. So I'm actually at a point when I'm considering expanding the scope of the project to include things like wind energy. At the level of literary analysis, any device that functions at the level of symbols or signs is fair game for my analysis, although I've isolated case studies that tend to foreground solar and I'm finding wind symbols. At the level of intellectual history, I'm particularly interested in those thinkers who explicitly foreground their own definitions of energy, and this is where it's getting confusing. Do I begin with Aristotle, Leibniz, Diderot? I mean, there's so many possible starting places. I basically want to challenge myself to understand literary and philosophical production as the generation of new energies that are themselves derived from solar and wind powers. Such powers, again, I'm arguing, energize orientations or commitments to equality, justice, and emancipation. And I'm looking at several presses currently, including Duke University Press, Fordham University Press, SUNY Press, and University of Minnesota Press as possibilities. I'm just basically relying on what I cite, looking at my apparatus and being like, that those might be possible venues. But yeah, thank you. I'm excited to chat about it if anyone has any questions, and thank you for listening. And I hope everyone's well.
Thanks, Sean. That was really interesting. So for the audience, if you have any questions, please go ahead and put them in the Q&A. But I'll start with the questions. With a question, Sean, can you talk a bit more about the Francophone perspective, the French perspective, and what that perspective bring uniquely to to your theories versus potentially other kinds of perspectives that are not French?
Definitely, yeah, and that's a really central research problem or question I've been engaging with for several years. I can get really interested in a variety of different languages as well. Like I want to learn German and Arabic. There's several different potential traditions or bodies of work in different languages that could be useful here. But in also kind of recognizing my own limits of specialty, I've kind of been wondering about this question, and there seemed to be specifically in the 18th century this interest around energy, particularly in French philosophy, which had a really kind of intense relationship, I think, to poetic language. I think immediately of somebody like Diderot, and the effects of that, I'm still teasing out the historical effects of that. I think in the 19th century, the focus kind of went to poetry in a pretty profound way. And that's not to say that the question of energy was necessarily like relegated. It's just one that I think manifested in French poetics. And then in the 20th century, I think there's this really interesting kind of synthesis between those two movements, like whether energy is defined according to science or whether energy is defined according to poetics. So in the 20th century, there's a lot of intellectual and literary production that I think is really invested. It's just one of those words that I keep seeing in all of the texts I'm reading that seems undefined and also kind of like conflicting. There's so many different definitions of it that are used. So I'm trying to clarify that across history. And then there's the question of the French language too, which would, that would be another project, which is if there is something about the French language that in itself, I have no idea, but that's kind of where my direction or research is going a little bit, for sure. So thank you.
I have a follow-up question if nobody has anything in the Q&A. And that is, Sean, you talked a bit about, the sort of concept philosophically about, non-humans being irrational, and then possibly humans being rational, right? And this kind of this Western philosophy of that. So can you sort of expound more of your thoughts about humans being irrational versus being rational and how that may impact this look about the apocalyptic sense that we have about about energy and climate?
Yeah, definitely. I'm really interested in that question, and specifically the effects that does relate actually in some way, I think, to the question of language, because I do think that so recently I've been teaching a book called, well, the translation is Eve Out of Her Ruins. And this is a Mauritian postcolonial novel, but the way that the language is kind of, I guess, distorted or affected in a certain way by, for example, like environmental trauma in particular, is something that really interests me. So I guess the question would be how, according to certain, like, given understandings of what is systematization in particular, and coherence and consistency and reason, I'm interested in understanding how those elements, for example, consistency, coherence, and systematization, manifest in poetics. So, for example, a book of poems that I'm looking at by Atel Adnan, who's a Lebanese American poet, is very disorienting and challenging material, but it's also a direct response to the traumatic legacies of the Lebanese Civil War. And so there's this question of, and I've been trying to read this with my students and trying to understand if there is a kind of logic or coherence or consistency to the poems. And I think there totally is. It's one that relates to legacies and histories in French poetry and philosophy as well. So yeah, it's a really interesting question. And yeah, I think that even those terms are kind of fair game for further definitional clarity, right? Like something like reason and consistency. Like I think those terms are would be interesting to kind of question, I guess.
Great, thanks, Sean. Thanks for those great answers. We're going to move on to our next presenter, and that is Kimberly Jenkins, and she will be talking about her dual language learning project. Kim.
Thank you. Hi, everyone. Thank you for the opportunity to talk a little bit about the work that I have been doing, my current work and my emerging line of research here at UNC. In general, my research focuses on language acquisition and language development in dual language learners. And by dual language learners, I'm referring to children who are in the process of acquiring 2 languages. And to share some information about dual language learners in the US, we know that about 20% of individuals ages 5 years and older in the US speak a language other than English in the home. And approximately 16% of school-age children in North Carolina speak a language other than English. And the largest subgroup of children are those who speak Spanish and English. So we know that there is existing evidence. So what we know currently is that Spanish-English dual language learners learn 2 languages with distinct grammar, and that the development of their language is a dynamic rather than a linear process. And there's currently a growing body of evidence that's focused on the grammatical skill of Spanish-English dual language learners. However, most of the work that's currently been done has come from cross-sectional studies. So the existing evidence that's based on longitudinal and concurrent work for the development of grammar in Spanish-English dual language learners remains somewhat limited. And so this creates a narrow empirical base that's focused on normative language development in Spanish-English dual language learners. As a result, determining when difficulties reflect typical, different, or impaired language patterns of language development can be challenging. And without this, this information, this evidence, this data, it's pivotal for being able to successfully diagnose and provide intervention. And without this information, dual language learners with language impairments are vulnerable to ongoing risk of language difficulties and reduced literacy skills. So my current work, the Dual Language Learning Project, represents a first step toward advancing diagnosis and intervention of language impairment among Spanish-English dual language learners in the United States. And the Dual Language Learning Project is part of an emerging line of research with the long-term goal of improving clinical efforts to differentiate language impairment from differences, to guide the development of least biased assessment and culturally sensitive intervention approaches, specifically those that are targeting the needs of Spanish-English dual language learners. So the short-term objective of my current work is to identify longitudinal patterns of normative development in English and Spanish grammar of Spanish-English dual language learners without known language impairment. And I'm looking currently at children from preschool through 1st grade. And we currently have 40 Spanish-English dual language learners in the project. And again, these children are not known to have a language impairment. We know that they have a variety of language exposure with speaking, a variety of extents of being exposed to English and Spanish in the home. And we're looking at around 400 narrative retail language samples for these children in Spanish and in English, and currently coding these language samples for the use of various types of grammatical features, forms, and categories in English and Spanish. And we are currently using growth curve modeling to estimate variability and to really be able to examine patterns of change that are occurring between and within participants over time. So in the future, I am planning to continue to explore longitudinal patterns of development in dual language learners And along with looking at children who have are not known to have language impairment, also planning to collect additional data to include children who do have language impairment. I'm also working towards developing and implementing developmentally appropriate modifications for existing cognitive assessment tools, such as tasks that are focused on verbal working memory. And I'm also interested in examining clinical treatment outcomes that are associated with the use of multi-component intervention plans, such as incorporating professional actions and partnerships with pertinent individuals, such as teachers and parents who can also implement these clinical actions. And so an example of such a program might incorporate parent and teacher training that supports developmental and maintenance of language skills in a heritage language. And I just would like to thank my research mentors and collaborators, as well as the participants, the dual language learners and their families who've taken part in this research, and acknowledge recent funding from NC Tracks to complete this work. Thank you.
Thanks, Kimberly. That was really interesting. I'll start with the first question. I wanted to expand a bit more about some of the work, I guess, that you might be going into the future on dual language learners that may not have difficulties in learning, or are there dual language learners that are, because of having the dual language, are actually excelling at learning in different ways? And can we learn from that and, in their interventions for those that might be struggling?
Sure, definitely. There are definitely dual language learners that don't experience any difficulties. So what I'm really trying to capture with this project currently is to know what patterns do we expect to see in normal language development over time. And we know that just in the process of learning both languages, there can be just different patterns that are, I would say, developmentally appropriate as children are learning their language. But there can also be a lot of variability and skill. And so we're really trying to, over this, in this project, trying to capture knowledge and understand better what patterns is it that we would expect, and being able to use this information then to compare to children who are learning their languages as dual language learners and do also have a language impairment, to see again, what are the differences that might exist, and what are the signs of difficulty versus patterns of that are associated with just learning two languages simultaneously or sequentially.
Mm-hmm. So a question in the chat here is, can you define more what you mean by language impairment? How is it typically defined?
Yeah, so with language impairment, it's the case that when we look at the domains of language, it can be the case that some children have a slower rate, or they have some challenges when they're learning vocabulary, or maybe when they're learning grammar. So it can extend to pragmatically too. So maybe when with turn taking, so it can be expanded across or exist across several domains of language, or it can be more specifically centered on one aspect of language.
Okay. And Terry Terry, do you want to just ask your question since you're on the?
Go ahead.
Panelists? Oh, okay. Terry's question was, how do you correlate your work with postnatal brain development?
So I haven't, well, I guess I would say that currently the project that I've looked at, I've looked at an earlier project and kind of do plan to revisit it, but I have looked at working memory, and I've used some behavioral tasks. And that specific project was looking at internal factors, as well as external factors. So internal factors, I was looking at verbal working memory, and then in a group of dual language learners. And then also for external factors, I was looking at the use of language exposure in the home and looking at children's grammatical performance and looking to see specifically in that study if there was any relationship between their grammatical performance and the variation in their verbal working memory performance and their language exposure in the home. So I would say that those are, that's like the beginning stages of that, trying to make that correlation between cognitive performance and cognitive ability, and then their language performance.
Thanks, Kimberly. We're going to move to our next presenter, who is Musamanga, and he's from the Department of Environmental and Engineering Sciences. And the title of his talk is, How Well Do Sanitation Technologies Intercept Release of Pathogens to the Environment? Some evidence work from Tamil Nadu in India. Musa, go ahead.
Can you hear me? All right. Thank you so much, Joyce. Can you see my screen?
Yes.
Cool. Okay. Thank you so much, Joyce, for the introduction. Today I'll be speaking about something that's quite interesting. So I am I'm a Carolina Postdoc Research Associate from the Department of Environmental Sciences, and my research focuses on planning, monitoring, and improvement of sanitation and human excreta management practices in the underserved communities, with an aim of promoting public health and well-being. So today I'll be speaking about something very small of my research, and I'll be talking about how well do these sanitation technologies intercept release of pathogens to the environment. And I use some of the evidence we've actually have from Tamil Nadu, India. So I'll start with the introduction. So globally, access to sanitation is an increasing challenge, especially in the underserved communities or marginalized communities, with approximately 700 million people practicing open defecation and 2 billion people lacking access to basic sanitation. And most recent research has showed us that inadequate sanitation and exposure to fecal pathogens is responsible for a substantial portion of global burden of disease, affecting about 2.2 billion people annually. So with this, the international community with the World Health Organizations have actually come up with an indicator of safely managed sanitation services for tracking the SDG 6.2. But this calls for safe management of human waste from the containment part to disposal. If you look at an example of Chapel Hill, we've got a sewer system. So everything, once you use a toilet, you flush everything, it goes through the sewer to the treatment. So this calls about safe management of waste throughout that chain. Whereas if you look at places like some of them, like in Chapel Hill, which are in underserved communities like Greensboro, they do not have sewers, so they have to use on-site sanitation systems. And these have got a bit of containment, emptying, transportation. So when the thing is full, it has to be emptied and conveyed for treatment. However, our most recent research has actually showed that the front part of the chain is where most pathogens are released. And therefore, we should focus on there if we are going to maximize public health benefits from the sanitation chain. And based on that, the problem is we do not know even how these systems perform. And that takes me to the question for today, is how does the daily pathogen release to the environment from these systems, from the liquid discharge or periodic dislodging, vary by technology? So in order to answer this question, we had a chance to go to India, and that's the place we went to in Tamil Nadu, India. And we had a chance to interact with community members. We conducted some kind of key informant interviews to understand the practices they are using for managing their human waste. I think I've shared the wrong screen. Can you see my slide, or are you seeing my notes?
We see your slides.
Okay, cool. Not my notes.
No.
Okay, cool. So what we did is we did some kind of community transect walks. So we had a chance to really find the kind of technologies they have in these communities, and also did some kind of structural observations and sanitation user survey to know how are they using these systems and how are they managing their human waste. So we had a chance to know what kind of technologies they have and had to collect samples from them. So some of the technologies we came across in these communities, hold on. Some of the technologies we came across in these communities, they had some kind of pattern of line pits, and this is how they look like. They are exfiltrating from the sides and the bottom. The others had fully lined walls, but the bottom was exfiltrating the liquid out. And the others had fully lined tanks with any kind of liquid discharge. And the others had a kind of a containment system, but a kind of a drain field, whereas some others had containment system, but discharging to open drains, flowing through the communities. And others actually did not have any containment system. Everything was going to the environment. These kind of systems you see here are not unique. They're actually found all over the world. Even in the US, people with this kind of systems, but we don't know how they perform. So what we actually had to do is to sample them to know how they perform. And we did a mass balance. So what we had to do is to know what are the number of pathogens coming into the system, what are the pathogens dying into the system, what are the pathogens that are released in the liquid discharge, and what are those that are emptied when the thing is full. And we also looked at some of them that are exfiltrating from these systems. However, today I want to talk about the pathogens that are actually coming from the liquid discharge and the ones that are being dislodged. So let's talk about the results. What did we find? So what I actually found in this figure, you see on the x-axis, we've got the different sanitation technologies. On the y-axis, we're having their kind of discharge. And these are discharged in the, these are discharged in the dislodging, the one that's the liquid discharge overflowing to the environment. What you can actually see directly is the sewers, the sanitary sewer and the direct pipe discharges recorded the highest pathogen or E. coli release. And we realized that these kind of fully lined tanks, which had overflows, actually had lower releases. And this showed us that reduction from these to these results into 1 to 2 log reduction. Where you see not applicable, it means those systems did not have a liquid discharge. And that's why they didn't have, that was not applicable to them. But another thing we actually found in here, we actually found that there was kind of a meaningful association between the retention time and the E. coli release. And looking at the design of this system, we realized there are kind of plumbing errors, pipe configurations that had a significant impact on the number of E. coli released. But then we went ahead and looked at what is being released by dislodging when the thing is full, what you take out. What you see here is the ones with not applicable do not have containment, like a sewer system doesn't have containment and direct pipe. But these you see here are the ones with containment. And what we actually realized that all these kind of systems, like the partly lined pits, which had self-containment for a longer time, performed better. They had the lowest E. coli release. Whereas we've actually found that even when you look at the storage periods with E. coli release, there was a significant association, meaning that the management of this waste should focus on how safely it's stored in this kind of containment system. But also we actually found that self-containment of this reduces the public health, the threat to public health than fresh content from non-containment. So we compared the E. coli release in the liquid discharge with that is in the solid discharge, and we found that the liquid discharge releases about 65 times more E. coli than what's released in the solid discharge. And that was something that we actually came across. And that means the sanitation planners or people should focus on how safely they can manage the liquid discharge than focusing on the sludge alone. And combining E. coli release from both the liquid and the solids, we actually came up with this kind of interesting figure, which shows on the x-axis the different technologies and the y-axis the E. coli release, and all the different performing technologies. What you could probably still see here, that the partly lined pits actually had the lowest performance. And when we went ahead to look at community toilets, which were actually the other containment system, they had the highest, the lowest performance because they released the highest pathogens. And this was because of the short duration period, the higher user practices, wrong emptying practices, which tells us that all people who are using this system, they should focus on how they operate them in a better way so as to minimize on equalized release or maximize public health benefits. And summarizing this, we actually found that storage periods played a huge role in minimizing the equalized release from these kind of systems. What are the takeaways? One, safe management of shit matters if you're going to maximize public health benefits in these underserved communities. Two, even if we talk about containment, the liquid discharge from these systems is important. As much as the World Health Organization promotes or talks more about kind of management of, sludging, we need to also look at the liquid discharge. And I think we also found that the public pipes and sewers overflowing to the environment act as epidemic needles. And therefore, we should not focus on only extending sewers everywhere, because look at having a sewer and it's kind of blocked and overflowing to, a kind of a road. They're releasing high pathogens. And lastly, self-containment of human waste is safe and actually could help minimize or maximize public health benefits. Next steps, we are developing this kind of pathogen flow diagram, which is going to help prioritize sanitation interventions to maximize public benefits in communities. I acknowledge some of the funders of the project and other people we've worked with all the way in Trichy and around. Thank you so much.
Thanks, Musa. That was really interesting. We have a question from the audience member. Do you partner with any institutions or organizations to conduct your research in India?
Thank you so much. That's a great question. Yes, we are partnering with some of the institutions in India, like IHS, the City Corporation for Trichy, and government officials are working with us on some of this kind of work. However, the next thing we want to also try to work on this, because this problem is also happening in the US, in the marginalized communities, and we're trying to connect with different communities to really help them solve these problems.
Thanks, Musa. We're actually running a little late on time, so I'm going to move on to our next speaker. And that's Ganga Bay, who is from the Department of Epidemiology. She'll talk about identity, aging, and health, a novel conceptualization. Ganga.
Wonderful. If you give me just one moment to share my screen. Okay, here we are. There we are. Okay. Are you all able to see my slides?
Yep.
Okay, wonderful. So thank you for the introduction, Joyce. And I'm happy to be sharing with you a very brief summary of my current research directions. Given the time, I will just jump right in. So recognizing the well-established relationships between neighborhoods and health outcomes, my research really seeks to understand, like many, how specifically our living environment gets under the skin, influences health and produces disease, and furthermore, whether there are ways to buffer the effects of those stressors while we do the very challenging work of deconstructing the structural foundations of health inequity. So one prominent theory is chronic stress and its effect on the body. And keeping this concept in mind and trying to tackle these two questions, I was drawn to an understudied area of epidemiology, psychosocial influences on aging. And bringing together theory from a number of disciplines, I have developed a new conceptual framework and proposed a novel hypothesis that there is a particular aspect of identity which makes individuals more or less susceptible to the physiological effects of environmental stressors. So to briefly summarize my novel identity, vitality, pathology framework, structural inequities are the origins of all pathways influencing differences in healthy life expectancy across populations, whether through shaping cultural paradigms or the neighborhood conditions that affect health behaviors, the expression of genetic traits, or the rate of aging. The ubiquitous nature of structural inequity means that no exposure outcome is untouched. And there's a lot of dense theory to get into here, but I really want to emphasize the primary distinction between my framework and the ecosocial framework on which much of the theory of social determinants of health is based, is this piece on the role of identity and psychosocial characteristics in contributing to health patterns across different socially positioned groups. So basically, I am arguing in this framework that an important and often overlooked factor in the mechanisms by which inequity is embodied to produce health and disease disparities is how identity states, or the modifiable ways individuals conceptualize their identity, the value of human beings and their own worth, influence how individuals appraise and cope with stress. Within this framework, the psychosocial characteristics associated with identity can promote resilience or susceptibility to the physiological effects of stress caused by structural inequity. And what I'm interested in using my framework to understand is what this means for disparities in cardiovascular and cognitive health. So in applying my IVP framework to epidemiologic research, this is the question that I'm trying to answer. Although a measure of identity state is under development and not yet available, I am currently examining whether biologic age really mediates the effects of neighborhood disadvantage on cardiovascular disease and cognitive decline, and whether this mediation is moderated by psychosocial resilience and risk factors that are theoretically predicted by identity. So to address this question, I am using data from two longitudinal cohorts, the ERIC study and the Jackson Hart study. And what we found so far has been really fascinating and novel. Inward anger expression accelerates cognitive decline. There was an up to 35% faster cognitive decline over a 10-year period, which is essentially 3.5 years of additional decline every 10 years for those in the most compared to the least deprived neighborhoods. But really that effect was only seen amongst those with the highest degrees of internalized anger. Secondly, that the more optimistic one is, the lower the risk of heart failure. And among those who were most optimistic compared with those who were least optimistic, there was a 12% lower rate of heart failure associated with increasing neighborhood disadvantage. 1 unit of neighborhood disadvantage. And then lastly, that risk factors, but not resilience factors, moderated that biological age pathway. So among those with negative affect scores, 2 standard deviations above the mean, there was a 22% increase in the rate of heart failure for every standard deviation increase in one of the measures of neighborhood disadvantage. And this again is for the effect of neighborhood disadvantage that is acting through increasing biological age. And so in conclusion, really, these preliminary analyses supported hypotheses from the IVP framework and indicate potential additional and novel intervention targets for environmental stressors on health. And my next steps will really focus on determining whether identity state as defined by my IVP theory does indeed predict the psychosocial factors that modify the effects of neighborhood stressors on health. And ultimately, my goal is really to do some intervention development and see if we can sort of develop population level interventions on these identity characteristics. So with that, I would like to acknowledge my funders, my mentors, and a very special thank you to the statistician who helps with these very complicated analyses. And I will take any questions.
Thanks, Ganga. It was really, really interesting. I know we had talked a bit about earlier, but maybe you could expound a bit more some conversations we had before about the differences in optimism and the differences in neighborhood deprivation that you've seen. So is optimism and results different in deprived neighborhood versus not deprived neighborhoods, or not so deprived neighborhoods?
Yeah, as I said before, a really interesting question and something that we definitely plan to look into more, because what we are finding so far is that these effects and the relationship, the modifying role of optimism really is only present in the most disadvantaged neighborhoods. And I think really, again, as I was saying before, I think it has a little bit to do with competing resources. So in these very deprived neighborhoods, I think the individual characteristics like people's levels of optimism or their depression or negative affect, I think have a greater impact because there are there are much fewer resources and other ways of positively coping. So that's my theory about why that might be happening, but it's certainly something that we have to explore more because we are finding that operates only within these really deprived neighborhoods.
Okay, and just a couple of questions on how you define inward anger and also how you define disadvantaged neighborhoods, or how it's quantified.
So we had a number of measures, and I'll answer that in out of order. But we had a number of measures for neighborhood disadvantage. So we use validated National Area Deprivation Index, for example, which is a very widely used measure of neighborhood disadvantage that essentially assigns a score for every census tract in the United States, where 1 is the least deprived and 100 is the most deprived. We used other measures like perceived physical disorders. So participants reported whether their neighborhoods were really noisy or had a lot of traffic or garbage around. And then for inward anger, we use validated scales. Well, the Jackson Hart study uses validated scales for all of their psychosocial measures. And for inward anger, that was evaluated using the state trait anger expression scale. And really, it's asked questions like, when you are angry, do you just sit and simmer in your anger, or do you let it out? And so questions like that, that kind of assess what people do when they're angry.
Great, thanks, Ganga. We will move to our next speaker, who is Julian Rucker from Department of Psychology and Neuroscience. And his title of his talk is Structurally Unjust: Lay Beliefs About Racism and Responses to Racial Inequality in Criminal Justice. Julian.
All right. Let me share my screen. Okay, are you seeing just the slides?
Yes.
Perfect. All right, thank you, Joyce, for that introduction. I'm Julian Rucker. As Joyce said, I'm a CPPFD postdoc in the Department of Psychology and Neuroscience. And generally, my research focuses on beliefs about the structural nature of discrimination and perceptions of racial inequality. And I'm just today going to give kind of a brief overview of some of the recent work I've been doing in that area. But to start, it certainly comes as no surprise to many of you, but stark, persistent racial disparities remain a hallmark of American society. And some of the most alarming disparities, which a lot of my work in general and the work that I'll talk about today will focus on, are those found in the US criminal justice system. So take, for instance, the fact that although African Americans make up only about 12% of the national population, they make up 33% of the national prison inmate population, and Black and brown people are overrepresented in just about every phase of our criminal justice system. So in my work, I build on kind of a long tradition of social science in that I've been interested in understanding how people respond to this sort of evidence of vast societal racial inequality. And, you know, as a psychologist, there are a lot of, surely a lot of different psychological factors at play here, but over the course of my research, I've been particularly interested in investigating whether responses to racial disparities, to this sort of evidence that you're seeing right now, may depend on how racism itself is conceptualized. And I'm particularly interested in whether it's conceptualized largely in terms of interpersonal biases between people or structural factors that disadvantage members of, or advantage members of particular racial groups. So to address this question, some of my work has looked at kind of large scale, nationally representative survey data. So in one study, for instance, we analyzed responses from Black and white American participants drawn from a nationally representative sample. This one was from CNN and the Kaiser Family Foundation. And we have found that racial differences in beliefs about interpersonal or structural nature of racism help to account for differences in perceptions of racial inequality in our criminal justice system. So more simply, we found that white participants' lower tendency to perceive racial inequality in the criminal justice system was explained by their greater tendency to endorse a more interpersonal rather than structural understanding of racism. So in addition to looking at national survey data, we've also conducted some experimental studies that test whether these lay beliefs influence subsequent responses after actually seeing racial disparity information rather than just kind of thinking about it in the abstract. So for example, in a set of studies, we looked to see whether lay beliefs about racism predict responses after exposure to evidence of racial inequality in the criminal justice system. And in these studies, we measure participants' lay beliefs with a measure like this. Participants are asked to rate how strongly they agree or disagree with statements in line with a more structural understanding of racism or a more interpersonal understanding of racism. And then we give them a lot of demographic information about the prison system, but in adapting from some work by Jennifer Aberhart and her colleagues at Stanford, we manipulate the racial demographics of the prison system such that they're either shown a relatively more modest or stark racial disparity in the national prison system. And then participants are given some basic information about harsh criminal justice policy, like a habitual offender law, or more commonly known as a 3 strikes law, sort of how it works, how prevalent these types of laws are in the US. And we generally find that among those with a more interpersonal understanding of racism, they tend to show more support for harsh policies, especially when they're shown starker disparities in the prison system. But among those with a more structural understanding, they show less support for harsh policies in general, and tend to be unaffected by disparity information, or at times show the opposite pattern of effects. They show less support when they see kind of evidence of starker disparities. So in follow-up studies, we found several other factors relevant to this link between structural racism beliefs and responses to racial inequality in the criminal justice system. For one thing, it matters how much people are concerned with the existence of societal inequality at all, or sort of how egalitarian they think society should be in general. So in several studies, we find that it's really people with a more structural understanding of racism and more egalitarian preferences. So folks who think that society should be less stratified by racially or by a number of different groups, whom seeing evidence of racial inequality in the prison system leads to less support for these really punitive criminal justice policies. Another relevant factor seems to be whether people are actually exposed to information about disparities. So, for example, in a follow-up study, we manipulated the racial demographics of the prison population such that participants were either shown demographic information or nothing. And generally we find that structural beliefs and general egalitarian beliefs most strongly predict policy support when people are actually shown disparity information versus sort of not seeing that sort of information. And for things that are kind of on the horizon, we're beginning to explore the relationship between lay beliefs about discrimination and perceived inequality for different target groups and in different contexts to see if it's a general phenomena or if it's more specific to racism and criminal justice. As an example, in another line of work, my colleagues and I have found that Americans consistently and pretty dramatically overestimate progress towards closing racial economic gaps in the US over the past several decades. And currently I've been starting to explore how these misperceptions may be informed by different lay beliefs about the structural nature of racism. I also have some forthcoming work looking at implications of beliefs about structural discrimination against one's own group for intergroup solidarity among members of different marginalized groups. And in future work, I also want to explore the educational and socialization processes, be it parental or community socialization, that help to develop beliefs about structural racism. And I'm curious too about the psychological motivations that impact these views. So some of my ongoing work that started with my dissertation looks at how beliefs may shift as a function of concern about personal merit, or that basically your accomplishments aren't attributable to your own hard work. And so with that, I'd like to thank my funding sources, my many wonderful collaborators, family and friends who've all supported this work over the years, and to all of you for listening.
Thanks, Julian. That was really interesting. So I'll start with a question. Julian, have you done studies where instead of showing, demographics data, showing a story or pictures, and how that affects people's perceptions?
Yeah, I'm trying to think if I have. We've played around with a couple of ways of conveying this information, either like by showing just raw data, by kind of putting in vignettes like you're talking about. And what we're finding in general is it's kind of hard to nail down, and it's sort of part of the effect. It's hard to nail down how people will make sense of that sort of information, because we really have to better understand the attributions they're making for that information. So if we show people that there are these vast disparities in these domains in terms of their interest in like supporting policies to reduce it, or even in the case of criminal justice, actually supporting harsh policies that can often make it worse, their attributions for the causes of those disparities really seem to matter much more than how we frame them. Or how we present the information in that sort of way, and whether it's graphs or vignettes so we've played around with that a bit, but that's definitely one of the bigger works in progress there.
Great, thanks, Julian.
Mhm.
I just want to give time to Sunai, so we'll move to Sunai's talk. So our next speaker is Sunai Udbarik from biology, and his title is Elucidating Ecological Complexity Across Scales.
Can you see my screen?
Yes.
Okay, fantastic. Okay, welcome everyone. I'm going to try to be as optimistic as Ganga suggested in her research. So I'm Sunai Udbarik, and my work is really focused on this question of species coexistence. And that's really, it's a question that we're pondering ever since Darwin, or even before Darwin. If you look at the tropics, or if you look at the microbiota in your guts, we have all this diversity, but we can't quite explain how this diversity emerges. And this is a lingering question, which I hope to resolve and win the Nobel Prize by the end of my lifetime. But anyhow, so much of my work is focused on this interaction between the ecology and evolutionary feedbacks. And increasingly, I've been interested in how these interactions are mediated by the microbiome. And there is lots of evidence that the microbiome plays a significant role. And a lot of this is based on sequencing technology, but we're kind of lacking what I call a theoretical framework that can explain this question of species coexistence, that can explain how communities, be they in the soil or the gut or the tropical rainforest, are structured. And so I'm particularly interested in how we can translate those interactions to these population levels. So here, a note, one of these circles can represent a species, and the arrows indicate what the interaction is. Okay, so, and trying to understand how these bottom-up effects scale up to structure these populations is what my research is driven by. Okay, so I'm gonna kind of update you on work that I started since coming here at UNC. And one focus of this work has focused on the evolution of virulence. And what we mean by virulence essentially is how parasites can induce host mortality. So, we oftentimes distinguish between different stages of parasite infectivity. So you can imagine parasite transmission being very important, but just because you have the parasite or the pathogen doesn't mean necessarily that you die, because there is host immunity. But here I'm specifically talking about virulence and trying to understand how virulence might evolve based on these ecoevolutionary feedbacks. And in particular, I'm interested in multiple infections. A lot of studies traditionally have focused on single host, single parasite interactions. And my work is trying to scale it up, but trying to look at how multiple infections play a role. And one approach in sort of tackling this question is using the power of experimental evolution. So we can imagine that we have hosts, in this case, these oval-shaped pink colors. And on the host, we have various microbes depicted by the various shapes and colors. And we can start at the first generation and then passage that community to the next generation, and then to the subsequent generation. And essentially what we're trying to do is we're trying to compare the ancestral population, so generation one, with the evolved generation at, say, generation 3, and trying to understand how these parasites are evolving across host generations. And then with those parameters from those experiments, we can actually inform mathematical models. So these are population level models where you have a susceptible population, so that's an uninfected host, and then some hosts might have pathogen A, some might have pathogen B, and others are co-infected by both. And so when we look at populations, we kind of see this prevalence of some hosts having one pathogen and some others having multiple pathogens, and we kind of have to account for how these things evolve over time. Now, the specific system that I'm using that I started here at UNC and I'm kind of developing, and it's part of my research program, is this interaction between an insect host, a parasitic nematode, and the symbiont of the nematode. Okay, so I'm going to walk you through it. So we refer to these as tripartite interactions, or 3-level interactions. So you can have a parasite in the soil, okay? So lots of parasites in the soil that seek hosts using chemical cues. Now this particular parasite also has a bacterial symbiont. So this is a tightly evolved mutualism between the bacteria and the parasite. And at some point, the parasite is able to find a soil-dwelling insect that it invades and starts the infection process. Okay, and as that continues, the bacterial symbiont of the nematode, of the parasite here, is being released in the host. So it's actually the bacteria that does the killing of the host. It has these virulence factors, these toxins that essentially kills the host. At the same time, the parasite is able to go through its entire life cycle. So it's benefiting from this mutualism. And after some point in time, the newly emerged progeny gets out of the host and starts seeking new hosts. So what I'm particularly interested in is, well, what if we add a second parasite? And you can imagine you kind of have this trade-off between having an additional competitor that also has its own bacterial symbiont. And there's this kind of this trade-off between within host interactions and between host interactions, okay? By which I mean transmission. And in addition, we really have to account for these other microbes, either in the soil or in the host, these sort of frequently associated microbes that can also play an important role. And so this is kind of the work that my program seeks to uncover, okay? And so the past summer, really, up to now, we have started to work on various aspects of the system with some of my undergraduate students, which I hope you can see on the right. I'll also show you some live parasites in action, okay? Very exciting, as you can see. And part of what we're trying to do is how these communities are actually structured. So we have been doing some work trying to disentangle the interaction between two parasites, okay? Steinernema and heterohepatitis. And what we essentially have done is change the order of infection as well as the timing of infection, okay? And so you can infect Steinernema early, you can infect heterohepatitis at a later time point, and you can look at the various combinations. So you have single treatments and co-exposed treatments, and you do it the other way around, where now the heterohepatitis parasite, you infected the host early, followed by Steinernema. And what we have found thus far is that we found higher levels of host mortality when the parasite heterohepatitis infects first, followed by Steinernema, okay? And in some next step, we're also analyzing some longitudinal data because we've actually been following this for a couple of months and try to see if that timing that sort of longer time span plays an important role. And in addition, we're actually looking at the bacteria and whether the bacteria of each of the parasites are mediating this interaction. All right. So we're kind of putting together a grant proposal. I'll just kind of outline some of the next steps. These were some of my collaborators. Based on some of this work, we hypothesized that interference competition, so competition among mostly unrelated parasites is more likely to lead to higher virulence or higher host mortality, and that this is mediated by their respective bacterial symbionts, okay? And so some of the follow-up work will focus on trying to actually quantify some of the nematode and symbiont fitness, okay? We can look at various fitness traits as shown in green. And then we also want to look how the bacterial symbionts are mediating some of this virulence. And lastly, we also want to look at the larger community of microbes, right? So I've mainly talked about these tightly evolved mutualists, but you can imagine that there are other players, so to speak, that are also important in this system. Then I want to acknowledge my cohort, of course, the Carolina Postdoctoral Program, my mentors, and the biology department. Thank you very much.
Thanks, Sunai. I think you're on your way to your Nobel Prize.
That was just a motivating.
Can you talk a bit about what you think the role of microbiota that may be beneficial, you know, how those interactions may interplay with each other? Just, you know, just theoretically, what your thoughts are on that.
Sorry, I missed the very first one.
You know, the interaction with microbiota that may be beneficial, or does that happen in the nematodes? I guess I'm not really familiar with how that works.
Yes, yeah, that's a good question. So yeah, so the microbiota is really happening at three levels: the insect, the nematode, the parasite, and so we haven't really disentangled the microbiota at each level, but you can imagine that each of them have a core microbiota that you'll always find in let's say, the gut. And those are beneficial because they help with nutrient depletion and so forth. Nutrient acquisition. But then there are other pathogenic microbes that are not beneficial, and so it's really a continuum, I would say, from mutualism to parasitism, and that can shift depending on the environmental conditions. And so we used to have this kind of very static point of view, and I've heard several of you talking about, you know, dynamic, you know, and I think this is a very dynamic system that can rapidly change depending on how favorable the environment is.
So, Sunai, you sort of answered the question that I had. In the lab, you can control the environment, but outside the lab, you can't. And so I would assume the environment has a huge impact on the results.
Yes, absolutely. I should say that these hosts are actual pests, so they have kind of adapted to this semi-natural environment. They are pantry moths. You might have them in your kitchen too. But one thing we can do is we can actually bring in, say, soil from soil collected environments and bring them into the lab, and so expose them to microbes that actually are in the environment. And so I think that's one way that you can mitigate it, but certainly true that you know, some of the growth media and the food media that these organisms are kept in the lab can bias your results. So ideally you have kind of a gradient of sort of a lab environment, but also a real environment where you can actually bring in things like soil or even different host genotypes. Okay, and that's the next step of this work. I'm actually going to put some of these insect hosts, rear them in the soil, and let the microbes get in there, and then bring them back to the lab and analyze what's happening with that community, and then compare it to lab strains.
Thanks, Sunai. So thank you all of our postdocs. These were great presentations, really, really interesting, wonderful work. And really fortunate to have all of you here at UNC doing it's really great and fascinating work. And so that closes and ends our session today. And I think this will all be recorded as well. So we'll distribute this out also to others who are interested in learning more about your work. Thank you.
Thank you, everyone.
Thanks. Have a good day.
Bye.
Thank you, folks.